Hello students, welcome to EPG Pathshala. I am Tanmay Datta, Assistant Professor of Biochemistry in the Department of Chemistry, Indian Institute of Technology, Delhi. Today, I am going to talk about DNA damage from the paper Molecular Biology, Genetic Engineering and Biotechnology. Today's lectures include how DNA is damaged by different kind of external and internal factors. We will also study about the different mode and nature of DNA damage. We will also learn how DNA damaging chemicals induce carcinogenesis. Therefore, the objectives of this module includes the understanding of different mode of DNA damage, how different kind of radiations induce DNA damage, mechanism of DNA damage by different kind of oxidative stress, some chemicals can serve as a carcinogen which induces the cancer. In this lecture, we will first study briefly about how DNA is damaged by different kinds of radiation of chemical compounds. We also focus how reactive oxygen species and other intercalating agents damage DNA. The proofreading function of DNA polymerases is essential for the fidelity of DNA replication and thus for the inheritance of correct genetic information to the descendants during cell division. Yet errors in DNA polymerization have been seen occasionally and may alter the nucleotide sequences of genes if not corrected. Chemical agents, either naturally present in the cell or in the external environment of the cell, can also alter DNA sequence. Severity of the damage in DNA determines whether the damaged portion can be repaired, transmit the intact genetic information, or can be repaired, leading to the loss of genetic information and sometimes cell death. Sometimes altered genetic information is passed on to the daughter cells because of imperfect restoration of gene sequences producing mutations. Genetic changes are usually notable only when they occur in germline cells of multicellular organisms, so that the change is transmitted to all the cells of the organism's offspring. Damage to the DNA of a somatic cell, in contrast, rarely has an effect beyond that cell unless the mutation contributes to a malignant transformation. DNA is a complex organic molecule and highly stable material which is required for the storage of genetic information. DNA is susceptible to spontaneous changes that would lead to mutation if left unrepaired even under normal conditions. There are three kinds of bonds in DNA which are potential for hydrolytic cleavage: phosphodiester bonds, N-glycosyl bonds, and bonds linking exocyclic amine groups to bases. In DNA strand, spontaneous phosphodiesterase bond cleavage is a very rare occurrence and probably does not make any significant contribution to DNA damage. However, an abasic site is formed upon N-glycosidic bond cleavage, which is also known as apurinic or apyrimidinic site. This figure represents how an abasic site is generated and how depurination leads to gene mutation. Reports show that DNA of each human cell loses about 18,000 purine bases, adenine and guanine, every day because their N-glycosyl linkages to sugar hydrolyze by a spontaneous reaction called depurination. In addition, reactive compounds generated from cellular metabolism can cause the deamination of DNA bases. The nature of the four bases of DNA makes the distinction between undamaged and damaged bases very clear. For example, every possible deamination event in DNA yields an unnatural base which can be directly recognized and removed by host repair system. a special situation occurs in vertebrate DNA where selected C nucleotides are methylated at specific CG sequences that are associated with inactive genes. The accidental deamination of these methylated C nucleotides produces the natural nucleotides T in a mismatched base pair with a G on the opposite DNA strand. This is another example of spontaneous base alteration in DNA. The figure shows the site of spontaneous alterations in DNA backbone. Red arrows indicate the target of oxidative damage. Blue and green arrows indicate the site of hydrolytic damage and alkylation. In this figure A, B and C, which shows DNA structure before and after UV radiation which lead to the distortion of DNA strand. Different high energy electromagnetic radiation in the environment including ultraviolet light, ionization radiation and certain chemical agents can physically damage DNA. For example, UV radiation in the range of 200 to 300 nanometer promotes the formation of a cyclobutane pyrimidine dimer between adjacent thymine residues on the same DNA strand to form an intra-strand thymine dimer. Similar cytosine and thymine cytosine dimers also form but less frequently. Such pyrimidine dimers locally distort DNA's base paired structure, interfering with transcription and replication. The kind of damage account for about 75% of UV-induced DNA damage. The cyclobutane ring is generated by forming one bond between C5 atoms and another between C6 atoms on adjacent pyrimidine ring. Structural studies shows bDNA can accommodate a single T-T dimer, forcing the helical axis to bend by about 30 degrees toward the major groove. The dimers 3' thymine can form normal Watson-Crick base pair with complementary adenine, and the interaction between the 5' thymine and its complementary adenine will be weaker than normal because a single hydrogen bond can be formed. Ionizing radiation also damages DNA either through its direct action on the DNA molecule or indirectly by inducing the formation of free radicals, particularly the hydroxyl radical, in the surrounding aqueous media. These can lead to strand breakage. The DNA damage produced by chemical mutagens, substances that induce mutation, falls into two major classes. One, the point mutation, in which one base pair is replaced by another one. These are further classified as transitions, in which one purine or pyrimidine is replaced by another, and transversions, in which a purine is replaced by pyrimidine or vice versa. The second one is insertion and deletion mutations, in which one or more nucleotide pairs are inserted in or deleted from DNA. This figure shows the formation of a radiation-induced cyclobutyl thymine dimer. Upon UV radiation of two adjacent thymine residues on a DNA strand forms dimer. Long covalent bonds joining the thymine rings are much shorter than the normal 3.4 angstrom spacing between staked ring in bDNA, which distorts DNA locally. So students, until this point, we have learned different sides of DNA backbone prone to be damaged and how radiation induces the formation of pyrimidine dimer. In a cell with impaired system, this kind of DNA damage can lead to cancer formation. We will discuss how reactive oxygen species or simply ROS damage DNA. Cellular respiration involves the generation of reactive oxygen species produced in electron transport chain, which probably do not cause any damage to nuclear DNA under normal physiological conditions. The reason behind these facts include a respiratory chain does not normally release these reactive oxygen species. Second reason, cells contain ROS scavenging enzymes like superoxide dismutase, which converts superoxide radical into molecular oxygen and hydrogen peroxide, and catalase, which converts hydrogen peroxides to oxygen and water. The third reason is superoxide or hydroxyl radicals, if released, are so reactive that they react with nearby biomolecules before acting on DNA. However, ROS produced by other mechanisms can cause considerable damage of nuclear DNA. The primary reason for this is the generation of hydroxyl radical from hydrogen peroxide, which is a normal metabolic product of many biochemical pathways, including one which converts guanine to uric acid. Hydrogen peroxides may escape the action of catalase and peroxidase and can be converted to a hydroxyl radical by the Fenton reaction. Different kind of products generated as a result of interaction of DNA bases with reactive oxygen and free radical species. Hydroxyl radical generated in Fenton reaction can cause many different kind of DNA damages, including oxidation of guanine to 8-oxoguanine and thymine to thymine glycol, etc. More than 100 different oxidative modifications leading to DNA damage have been reported till date. Deficiency in the repair of these modified bases results in transversion mutation. When a DNA strand with such kind of modified nucleotides is replicated, the OXO-G can base pair with either cytosine or adenosine, causing a GC to TA transversion. Thymine glycol has a cytotoxic effect on cell due to its inhibitory effect on DNA replication. In this section, we will learn how deamination of DNA bases lead to the generation of point mutation. Oxidative deamination of aromatic primary amines is one of the reasons of point mutation. Cytosine is thereby converted to uracil and adenine to the guanine like hypoxanthine, which forms two of the guanine's 3 hydrogen bonds with cytosine. Hence, deamination of DNA results in both AT to GC and GC to AT transitions. One example of such DNA deaminating agents is nitrous acid, which is formed from nitrates used as preservatives in processed meats such as bacon, sausage, hot dogs, etc. Despite its potential mutagenic activity, nitrite, the conjugate base of nitrous acid, is still used as a preservative because it also prevents the growth of Clostridium botulinum, the causative agent of botulism. Bisulfite, an additive that is sometimes present in wine, beer, fruit juice, and dried fruits, also significantly increases the rate of cytosine deamination without affecting purine or 5-methyl cytosine. A few cytosine bases in eukaryotic DNA are converted to the modified base 5-methyl cytosine. This modified base is located in the so-called CpG islands, which are small segments of DNA often present in regulatory elements termed promoters, the upstream sequence of transcription start site. The rate of spontaneous deamination of 5-methyl cytosine bases in CpG islands is even greater than that for cytosine. The product in this case in thymine resulting in the conversion of a CG base pair to a TA base pair. The figure shows the structure of different alkylating agents of DNA bases. The backbone of DNA consists of highly electron-rich molecules and therefore a target of attack by electron-seeking chemicals or electrophiles. Transfer of alkyl group such as methyl, ethyl, propyl, etc. to the electron-rich atoms of DNA by a highly reactive group of electrophiles lead to damaged DNA by alkylation. Alkylation takes place at nitrogen and oxygen atom external to the base ring system. Nitrogen non-bridging oxygen atom of the phosphate group and nitrogen atom in the base ring except those linked to ribose sugar. The example of such alkylating agents are dimethyl sulfate, which is frequently used in research laboratories as a mutagenic agent for random mutagenesis. Another example are nitrogen mustard, ethyl nitrosourea, N-methyl N-nitro N-nitrosoguanidine. The exposure of DNA to N-methyl N-nitro N-nitrosoguanidine yields, among other products, orthomethylguanine residue, which can base pair with either C or T. S-adenosyl-methionine, a metabolic methylating agent, occasionally non-enzymatically methylates a base to form a derivative such as 3-methyladenine and 7-methylguanine residues. However, base methylation also have normal physiological functions. The alkylation of the N7 position of a purine nucleotide renders its glycosidic bond susceptible to hydrolysis, leading to loss of the base. The resulting gap in the sequence is filled in by an error-prone enzymatic repair system. Transversions arise when the missing purine is replaced by a pyrimidine. In this environment, different hazardous materials in shoot is a mixture of polycyclic aromatic hydrocarbon, the abbreviated form is PAH, formed by the incomplete combustion of wood or coal used as a fuel. Tobacco smoke and Charboiled meats also contain similar type of PAHs. The common structure of different types of PAHs in two or more fused aromatic rings. These hydrophobic hydrocarbons are not able to damage DNA unless they are metabolically activated. Metabolic activation pathway requires the action of several different enzymes, which includes cytochrome P450, epoxide hydrolase, etc. However, during this enzyme-mediated activation process, DNA can be alkylated by PA. We will be discussing the effect of intercalating agents on DNA backbone. Intercalating agents are reported to have the capacity to mutate DNA. Insertion or deletion mutations may arise from the treatment of DNA with intercalating agents such as acridine orange, proflavin, ethidium bromide, etc. Intercalation of such a molecule in a DNA strand increases the, the distance 2 times between two consecutive base pairs. The replication of the distorted DNA occasionally results in the insertion or deletion of one or more nucleotides in the newly synthesized polynucleotide. Insertion and deletions of large segments generally arise from aberrant crossover events. A large number of reports regarding mutagenesis is that mutations as a result of DNA polymerase errors, spontaneous modifications, or chemical damage to DNA occurs at random. John Cairns explained this paradigm and demonstrated that bacteria which are unable to digest lactose preferentially acquired the mutations they needed in order to use lactose when it was the only nutrient available. This observation, which suggests that bacteria can direct mutations that benefit them, more likely reflects a non-specific adaptive response in which the overall rate of mutations, whether it is useful or non-useful increases when the cells are under metabolic stress. The activation of error-prone DNA repair and recombination systems that are relatively inactive in normally growing cells render the cell in a hypermutable state. In this section, we will learn how some mutagens can serve as carcinogens. Mutations in intergenic regions or in the intron sequence of a gene does not show any phenotypic effect. Similarly, the redundancy of the genetic code, more than one trinucleotide may specify a particular amino acid, can mask point mutation. Even alteration of amino acid sequence of a protein may not cause any change in its function if that happens in the surface loop without affecting the catalytic and accessory regulatory side. However, in some cases, a single mutation in some growth regulatory proteins of the cell can cause the dysfunction of that protein, resulting in unrestricted growth, which eventually leads to cancer. As many as 80% of human cancers may be caused by carcinogen that damage DNA or interfere with its replication or repair. Consequently, many mutagens are turned out to be carcinogens. There are presently over 80,000 man-made chemicals of commercial importance, and recent reports show that nearly 1,000 new ones are introduced every year. The standard animal tests for carcinogenesis, exposing rat or mice to high levels of the suspected carcinogen and checking for cancer, are expensive and require several years to complete. Thus, relatively few substances have been tested in that manner. Likewise, epidemiological studies in humans are costly, time-consuming, and often inconclusive. A rapid and effective bacterial assay for carcinogenicity was devised by Bruce Ames, which is based on the high correlation between carcinogenesis and mutagenesis. He constructed special strains of bacterium Salmonella typhimurium which lacks histidine synthesizing gene and hence under normal condition cannot synthesize histidine and therefore cannot grow in its absence. Mutation, if occurred in these strains, is reflected by their reversion to His minus phenotype. In the Ames test, nearly 10 to the power 9 test bacteria are sprayed on a culture plate that lacks histidine but contain a mutagen in appropriate concentration. Presence of mutagen in the culture media causes some of His minus bacteria to become His plus so that they grow into visible colonies after 2 days at 37 degrees centigrade. The mutagenicity of a substance is scored as the number of such colonies minus the few spontaneously revertant colonies that occur in the absence of the mutagen. Dose response curves, which are generated by testing a given compound at a number of concentration, are almost always linear, indicating that there is no threshold concentration of mutagenesis. About 80% of the compounds determined to be carcinogens in whole animal experiment are also mutagenic by the Ames test. Because many non-carcinogens are converted to carcinogens in the liver or in other tissues via a variety of detoxification reactions, as for example those catalyzed by the cytochrome P450. A small amount of rat liver homogeneate is therefore included in the Ames test medium in order to approximate the effects of mammalian metabolism. Alternatively, carcinogenicity testing may be carried out using mammalian cell cultures rather than bacteria. Some chemicals to which humans had been extensively exposed that were found to be mutagenic by the Ames test were later found to be carcinogenic in animal tests. They are tris 2,3-dibromopropyl phosphate, which was used as a flame retardant in children's sleepwear in the mid 1970s and can be absorbed through the skin. Another example is furyl furamide, which was used in Japan in 1960s and 1970s as an antibacterial additive in many prepared foods, and which has passed two animal tests before it was found to be mutagenic. We will be discussing here how cross-linking agents damage DNA. Many chemical compounds which has alkylating ability have two reactive sites, and therefore the action of those compounds on DNA can form intra-strand, that is within the same strand, or inter-strand connecting the opposite strand, crosslinks. Inter-strand crosslink prevent DNA strand separation and, if not corrected, are lethal. Some examples of such cross-linking agents are nitrogen master gas, which attacks the central nervous system, and cisplatin, sorelin, which is a naturally occurring substance synthesized by some members of the carrot plant family. The cytotoxic effects of all these compounds appear to be due to inter-strand cross-linking, which blocks the replication and transcription machinery. What we have learned in last few slides that some mutagens can serve as a carcinogen. Mutation induced by some mutagens can lead to cancer formation. Mutations in intergenic regions or in the intron sequence of a gene does not show any phenotypic effects. Similarly, the redundancy of the genetic code, more than one trinucleotide may specify a particular amino acid, can mask point mutation. Even alteration of amino acid sequence of a protein may not cause any change in its function if that happens in the surface loop without affecting the catalytic and accessory regulatory sites. However, in some cases, a single mutation in some growth regulatory proteins of the cell can cause the dysfunction of that protein, resulting in unrestricted growth, which eventually leads to cancer. As many as 80% of human cancers may be caused by carcinogens that damage DNA or interfere with its replication or repair. Consequently, many mutagens are turned out to be carcinogens. There are presently over 80,000 man-made chemicals of commercial importance, and recent reports show that nearly thousands new ones are introduced every year. Some chemicals to which humans had been extensively exposed that were found to be mutagenic by the Ames test were later found to be carcinogenic in the animal tests. They are tris 2,3-dibromophenyl phosphate, as described before, which was used as a flame retardant in children's sleepwear in the mid 1970s and can be absorbed through the skin, and furyl furamide, which was used in Japan's in 1960s and 1970s as an antibacterial additives in many prepared foods, and which had passed two animal tests before it was found to be mutagenic. So students, to summarize, in this lecture we learned about how different kind of radiations damage DNA and also the different mode of DNA damage, how damage in DNA generates mutation in some essential genes in a cell with impaired repair system, which is also described in detail. Mutagenesis in some essential gene can also lead to carcinogenesis, that is cancer. In this lecture, we cover some more important points about different physiological and environmental factors that causes DNA damage, how DNA bases are altered in the damaged conditions or different stress conditions, and how DNA damage induces mutations in a gene if the repair system is impaired. Mutagenesis of some essential genes can lead to cancer as described before. Formation or the generation of cancer can have very lethal effect, as you are all aware of. Thank you.